Hello everyone, welcome to this AlphaLife webinar. Looks like we're live now. First of all, I would like to just take a few minutes to get people into the webinar. So we'll start off with an intro question, then afterwards we'll go into the practical stuff and introduce you to your speaker and today's format. So first of all, Thomas, I know you just attended the PEPCA Wholesale Protein Conference virtually. At this conference, industry experts, regulatory authorities, and CROs meet up to discuss the world of HCP analysis. But can you maybe give us a few of your main takebacks from this year's conference?
Yes, yeah, I think we have attended this conference for the last 9 years, I think. And when we started 9 years ago, it was mainly people using ELISA. So it was 80% using ELISA and only 20% using mass spectrometry. But this is completely the opposite today. So 80% of the talks and presentations are about or including mass spectrometry and 20% ELISA. So there's been a big change in the way you look at the wholesale proteins.
Okay, that's interesting to hear that it has changed over the year. Is there any of the presentations you would like to highlight, or?
I think that this year there was some presentation actually giving info about feedback from regulatory authorities. That's quite interesting. That's not typically something we hear about. And I also think there was a lot of focus on looking at problematic HCPs, and that's also something that has come up with the introduction of mass spectrometry, that you now can look at specific wholesale proteins and you start to look at those even at very low level, because they can be of concern even below 1 ppm.
Okay, great. Well, it looks like people are coming into the webinar, so I think we'll get started with the practical stuff. First of all, once again, welcome to this AlphaLife webinar. We're so glad to have you all here today. And thank you for the massive interest in this topic and this webinar. Today's topic is LCMS for HCP assay validation and GMP release testing for complex samples such as vaccines, cell and gene therapy, and bacteriophage products. So for today's product, I'm your host. My name is Martin Rask Johansen. I work as a client relations manager here at AlphaLife, and I'll try to guide you through this webinar today. For today's topic, we have found our speaker in AlphaLife, among AlphaLife colleagues. It's Thomas Kofoed, the CEO and co-founder of AlphaLife. He has a PhD in organic chemistry and more than 20 years of experience working with protein analysis and mass spectrometry. So for today's presentation, Thomas will present around 25 to 30 minutes. Then afterwards, we'll use around 15 minutes for questions. So please, if you have any questions, you're welcome to put them into the chat. And also, if you experience any technical issues, please put them in the chat as well. Then our colleague Jette will try to sort them out. So I think we'll just share some slides, Thomas, and go into the presentation.
Yes.
It seems that people are online. I see a list of people now. Perfect.
So
go ahead, Thomas.
Yes, thank you. I also take this opportunity to thank you for joining today. As you can see, the title is related to GMP release assay of HCP analysis using mass spectrometry. And I think since we started developing mass spec-based analysis of HCPs, we have had an increasing number of clients asking us if we could provide this service as a GMP release assay. Of course, there's a lot of products where ELISA is perfectly suited and where you have good ELISA assays for monitoring HCPs. But especially within the more complex products, we hear from clients that they have issues with ELISA assays, and also people that are in a need for a fast development of a HCP assay. Then mass spectrometry is a very good alternative. So that's what I will talk to you about today, tell you about the methods that we have established here at AlphaLife and how we have controlled the variability of the assay. And then finally, I will also show you some cases. I think we have 4 cases, customer cases that are quite recent. I will just share the problems that they had and how we solved it with our solutions. So first of all, I would just give you a short introduction to AlphaLyse. So we are a mass spec-based company. So we have focus on helping companies that are developing new biologics, developing better analytics, so you can bring safe drugs to the market faster. And we have been around for more than 20 years. We have worked with a lot of different clients, mainly in Europe and North America. And within the HCP-based projects, we have had more than 350 projects by now, and still growing. And then since last year, we have also had a GMP accreditation for doing HCP analysis by mass spec. So this is how most of our clients look into the development of a new biologics. So typically our clients are either in preclinical or in phase 1, 2, and 3, but also we have an increasing number of clients that are producing for market. And as you perhaps all know, in the old days, and for most marketed biologics, we had ELISA only for monitoring host cell proteins. And typically what people were doing was using a commercially available ELISA assay early on for preclinical and phase 1 and 2. And then once they got into the later stages, they had to develop a process-specific ELISA. And that's been the strategy for most people developing or new biologics. But then with the introduction of mass spectrometry 10 years ago, approximately, an increasing number, and especially our clients, they are using an orthogonal method. So they are actually combining ELISA with mass spectrometry. And we also hear that the FDA and EMA is getting back to people just doing ELISA assays, that they should support their HCP data with mass spec data. So I think a lot of the new products, both monoclonal antibodies, but also other recombinant proteins, are doing this as a strategy, a combination of ELISA and MS. And then, of course, we have some projects where it's very difficult either to develop an ELISA, or you don't have the time to develop an ELISA. If you use a special expression system where you can't get the commercially available ELISA assays. And for those clients, and that's actually the ones that have requested that we provided this as a GMP service, they can use this strategy. We call it MS only, where you only are using mass spectrometry to monitoring your host cell protein. This is the US Pharmacopoeia. That's the chapter that most people are looking into when they are starting developing or looking at how to monitor host cell proteins. And until recently, this was the chapter that people were looking into, and it was mainly looking at ELISA assays. So not so much discussion about the orthogonal method and not much about mass spectrometry. But just a few weeks ago, there was an amendment to this chapter, 1132.1, which is saying residual host cell protein measurement in biopharmaceuticals by mass spectrometry. So by the introduction of this chapter, mass spectrometry has got an important role in monitoring host cell proteins. It's up for review right now, but at the end of July, it will be finalized, and hopefully then later this year, the final chapter will be published. So it's a big advance in HCP analysis that we now have this chapter in the USP. So as I mentioned, we have been doing this for the last 10 years, and we learned quite a lot about HCPs. We were new to HCPs when we started, and I will just share some of the experience we have had with mass spectrometry and the feedback we have got from clients over the years. So originally, we were mainly using mass spectrometry as an orthogonal method to ELISA to be sure that the ELISA was actually catching most of the HCPs. But as you know, with mass spectrometry, you just not get a number, but you also get identification of individual host cell proteins. And this opened up a completely new way of looking at risk assessment of your HCPs now that you get the ID of those host cell proteins. So you can do much more detailed risk assessment. And also with the detailed information about the HCPs, you can do process development in a more data-driven way. So we have a lot of clients using mass spectrometry to optimize their process and make decisions on optimizations based on mass spec data. And of course, we have a lot of clients looking at batch-to-batch variability, comparability. And actually, we are also developing mass spectrometry-based services for looking at ELISA reagents to characterize ELISA reagents and do coverage analysis. And in this part, we also have clients that have used it for IND documentation. Since we started, we have had more and more clients getting into the later stage phases, so we can see they use it a slightly different way. They use it especially when they scale up, or if they move from one production site to another. Again, then it's a perfectly suited tool for looking at what's going on. Is it the same HCPs I get in the new site? And we have people using it, especially for PPQ runs, where you run 3 GMP batches and then look at the HCPs using mass spectrometry. And again, it's providing data that are very convincing and can show that you have a stable and robust production platform. And then since last year, we can also do release testing under GMP, and that is what I'm going to talk to you about today. So I think it's important to tell you that we have actually the use of 2 different methods when we are doing our HCP analysis by mass spec. So we have what we call the HCP characterization, and this is the assay we have been using for the last 10 years. It's based on SWOT data independent acquisition, LCMS. If you're not an expert, you can watch one of our other webinars where we explain in more detail about this. But the assay is in principle a 3-step procedure. First, there's a sample preparation. In the sample preparation, we never look at the intact proteins. We also always do a digest, so the proteins are digested into peptides. First, they are actually precipitated, the proteins, and then we denaturate, alkylate, and digest. And before that, we actually add intact protein as internal standards. So we typically use a mixture of 7 different proteins that we add at a specific amount into your samples, and then we do this denaturation, alkylation, and digestion. And we actually managed to put the whole procedure into a robotic system, so we don't have any manual handling in there, and also adding to the robustness of the method. And once we have the peptides, we load them onto a LCMS system. It's a microflow LC system with a reverse phase column, and then you elute off the peptides into the mass spec instrument using a gradient of acetonitrile in water. And then we run the mass spec in this data independent acquisition mode for increased both sensitivity, but also to increase the accuracy in quantification. And this generates a huge amount of data. We get a lot of mass spec data, typically very big files, and we have to search those in specific databases. So for each project, we will set up a database with all the proteins that might be in the sample. So it will be, of course, the proteome of the organism, other proteins that might be added, trypsin, and also contaminating proteins. And then we search the data and we identify and quantify the host cell proteins in the given sample. So how come is it that mass spectrometry hasn't been used much in HCP analysis, and especially why is it that nobody has put it into a GMP environment? And I think I will show this by explaining a little bit about the variability of the assay, or the possibility for creating variability. First of all, we have the sample variability. You can imagine we get a lot of different samples, different drug substances, monoclonal antibodies, recombinant proteins, fusion proteins, vaccines, and cell and gene therapy products. So alone there, you have a lot of variability. And then we typically get samples that are from the early stages all the way to, not to early stages, but for not purified material or just after harvest, and then all the way to drug substance. So there is, of course, a big difference between these samples. And then we do the sample preparation. This digest, of course, you can imagine having different buffers, different concentration of proteins. It creates a possibility for variability. And then you use an enzyme, a protease, to cut the proteins. And also these enzymes can have different activities. And then you load these many peptides onto a ELISA system where you have a column. The column can be new, the column can be old. And then you elute off the peptide into a mass spec instrument where you again also have variability in the sensitivity of the instrument. If it's just cleaned, it has a higher sensitivity than it has when it's been running for weeks. And you generate huge data set that you have to search in databases using software. And on top of that, a lot of this is manual handling and evaluation. So you can imagine bringing this into a GMP environment without having control on the variability. You are, I would say, in big trouble. So I will just share with you how we have controlled the variability and also documented. So this is, we have taken the 7 standard proteins we have added in each project. We, of course, when we look across different projects, we can't look at the HCPs. We have to look at the standards. They are the same throughout all these projects. So we put all the data from these more than 150 projects, more than 1,000 samples, into a database. And when we look at the intensity of the 7 standard proteins over time, so you can see here we are in 2020, and in the end we are in end of 2022. So over 2 years we have monitored the standard proteins, and the intensity of those standard proteins are given here in this graph. And you can see it vary quite a lot. So if the instrument is just cleaned or if the project is running well, then you have a higher signal from the standards than you have when it's not cleaned, or for certain projects where the digest efficiency just is lower. So this is of course not good enough if you want to use it over time. I also have to say that these analyses were done on 3 different mass spec instruments and by different analysts. So it has all the variability that is part of what we are doing. So if we look at it this way, we have a big issue with getting it into a GMP environment. But when we then use one of the standard proteins for normalization, then the data looks much more uniform, and you can actually see that most of the standards are within what we call double or half amount. So they have all been, in this case, added at 2,000 ppm, and we are within the 1,000 to 4,000 for almost all of these projects. So by using the internal standard proteins for normalization, we remove this variability, and this is why we have been able to bring this analysis into a GMP environment. When we look at the standards here, we have again it's the same database. We have just highlighted the 7 standard proteins, so number 1, 2, 3, 4, and 5, and 6, and 7, and you can then see how they vary over all these different projects. So the CVs for most of them are below 20%, and the intra CVs below 10%. You can also see that actually this number 5 standard is always a bit higher than the others, and that's the fact of mass spectrometry, that sometimes a protein generate peptides that fly well in a mass spec instrument, and they will for sure generate a higher signal even at the same concentration as other proteins. So we kept it there because we know it's always in the high end. Actually, typically it's without then or outside the range we typically set between double or half amount. We have also looked at the database just to look at the different drug substances we have analyzed. You can see, of course, most of the drug substances are either antibodies or recombinant proteins, but we have also an increasing number of gene therapy products and also vaccines. Non-protein are typically small molecules where you are using proteins for the process, and then we have still not a lot of projects related to cell therapy. So a good mix of projects showing that the assay actually is very robust and can be used across different types of drug substances. And when you look at the CVs on these different drug substances, you can see for most of them, actually the CVs are below 6% for the standard proteins. When we look at those 7 standard proteins, it's somewhat higher for cell therapies, and we think it might be that the variability in that assay is, or the different projects have been quite different, but we would like to run more and learn more about cell therapies before we can rule out that it's still working with this assay. So as I mentioned, the first assay I explained was the characterization assay by this SWOT-DIA method. But for bringing the analysis into a GMP environment, we had to switch to an assay based on a triple quad instrument and an MRM method. The reason why we had to do this, I will come back to, but I'll just show you here that we got a certificate for GMP analysis last year, and we ran the first project where we released a COVID-19 vaccine for one of our clients using this assay, and they got approval and now are running the project under phase 3 and hopefully soon for market. So the assay is pretty much the same as the previous assay I explained to you, the characterization assay. The difference is not in the sample preparation. It's we still use the internal standard proteins as internal standards, and we still use a robotic system, of course now qualified to GMP level. But we do the denaturation, alkylation, and digestion pretty much or the same way as we did with the characterization assay. The difference is mainly related to the mass spec analysis, and this is actually the reason why we had to shift from the DIA SWOT method to the MRM assay, because we needed to have an instrument that we could qualify, not only the instrument but also the software. And we are working on also getting the characterization assay to work on a qualified instrument and with qualified software. But we are not there quite yet, but we hope to be able to provide that analysis also as GMP. But for now, it's based on a MRM assay run on a triple quad instrument. So we use a standard workflow LC system, and we are using this MRM acquisition on selected proteins. So with this assay, we have analyzed typically a number of samples and know which HCPs we are looking for, and then we select peptides from these proteins and set up a targeted method using MRM acquisition on a triple quad instrument. And then we do the quantification using these specific peptides. We still have this internal calibration for reproducibility using the standards, and we also have some standards that qualify the assay. So pretty much the difference is related to the MRM system that we are using. When we do the validation, we do this in accordance with the ICH Q2 guidelines. So we look at linearity, range, precision, also intermediate precision, accuracy, lower limit of quantitation, and specificity. So we make up, of course, a validation protocol and have this approved by the clients before we start the validation procedure. So now I will just share with you the learnings from running GMP assays for the last year and a half. First of all, I just want to show you this overview of the projects we have done so far. So we have divided them into still validation, but non-GMP validation, and those are done either on the SWOT-DIA LCMS method, or we can also do it if the GMP validation is not needed completely yet, then we can do it also on the MRM LCMS assay. And then the GMP validation is done on MRM LCMS, where we have used it for both HCP assay, looking at residual proteins, and also in one case where we looked at the viral protein ratio. And we are now also developing what we call platform assays. Those are specific assays for proteins that you can look at across projects, and especially problematic HCPs for monoclonal antibodies, lipases and esterases. We can set up a general assay we can use and validate and then use on an ongoing basis for our clients. And also related to monoclonal antibodies, we have an issue with protein A that typically needs to be documented that the level of protein A is kept at a certain level. So we can do both a non-GMP validation using both method and GMP validation only using the MRM LCMS method. And then, of course, for the GMP validated projects, we create a certificate of analysis. And the specification for that is something we are setting with the clients. But it's either total level of selected proteins, that's what we've seen until now, either in ppm or nanogram per ml. But it can also be on individual protein levels, again in ppm or nanogram per ml. So this is something we do in collaboration with our clients to find out what kind of data do we need on the certificate. So the first case I have here is a non-GMP validation HCP assay. This client, they actually produced bacteriophage products. They were in phase 2. They actually had 4 different bacteria. They had 2 gram negative and 2 gram positive, and several species per genus. They didn't have any ELISA available for any of the programs, so they had to develop either an ELISA or use mass spectrometry. They are in phase 2, so they went for the non-GMP validation, and also we did it on the SWOT DIA method and not the MRM. And the idea was to determine the total HCP level in those samples they provided to us. And here you can see the linearity. Actually, in this case, we used a standard protein to show linearity. So we had a calibration curve. So here we have added from 0 to 15,000 nanogram per ml. And we can show in those samples that the signal intensity for this standard protein is linear with regards to the amount we have added. And we ran the intermediate precision in 3 different days in triplicates, 2 triplicates. And as you can see here, the CVs are below 20%, almost below 10 for all of them. And we actually, in this case, looked at a low HCP, a medium HCP, and then 3 high amount HCPs to test the CVs with this assay. So a very, very good assay and easy to show that there's a linear relation between the mass spec data and the amount of certain HCPs. The second case was a GMP validation of an HCP assay. This client, they were developing a new vaccine using an unusual expression system. So they didn't have any ELISA assays available. They were considering developing an ELISA assay, but they could just look into the timelines of that and not even be sure that they would get an ELISA that came out with a positive result. So they decided to go with a mass spec based assay. They were also under time constraints, so they needed to develop the assay really, really fast. So they needed a fully validated GMP assays for phase 3 and for market. And we started out analyzing a number of batches with the standard characterization method to identify the host cell proteins in different batches. And once we have done that, we could see that all 3 batches only contained a maximum of 5 HCPs above a lower limit of quantification. So we selected those 5 HCPs and established the MRM method for those HCPs. And then we validated the assay within 4 months, and it's now being used for release of GMP batches. And we have a certificate we provide with all those batches, just saying the total HCP amount reported. This Third case is a GMP validation, not on host cell proteins, but residual proteins. And I think this is also a very good way to look at not only host cell proteins, but residual proteins that you typically use during downstream processing. In this case, it was a phase 3 product. We had to test, or they used 2 enzymes for site-specific modification of a recombinant protein. And they, of course, needed to show that those 2 enzymes were removed to a certain level in the final drug substance. Again, they didn't have any possibility to develop an ELISA assay, and they needed a fully validated GMP assay within 6 months for batch release. So again, we ran the SWOT-DIA analysis of the 2 enzymes to find peptides, suitable peptides for MRM method development. We selected a number of those peptides and then set up the method, developed the method, qualified and validated the method, and we could see in this case they were interested in to go down to 10 to 25 ppm. Typically with the MRM method, we can actually go somewhat lower, often below 1 ppm and sometimes even below 0.1 ppm with the MRM method. But for this method, it was suitable for just going down to 10 ppm. And as I mentioned, they would use it as a release assay for phase 3 and for marketed product. And again, here we had the possibility to spike in the enzymes because we had them recombinantly and in pure form. So I'm just showing here one of the peptides, the signal from one of the peptides with the MRM method, where you can see there is a full linearity all the way down to 10 ppm. And you can also see over here that the R-square value is really good, and those numbers are very accurate. So this assay is very, very good for quantifying those specific enzymes in those samples that they needed. Finally, the last case, case 4, was a GMP validation of a viral protein assay. Again, slightly different than just looking at HCPs, but this was a marketed product. They have been on the market for a year or so, I think, and they had 2 viral proteins of interest that they needed to find the ratio because it was critical for viral maturation. And the authorities actually requested that they, in phase 4, should monitor the ratio between these 2 viral proteins over the coming 2 years. So we developed the MRM method based on those 2 proteins. We found signature peptides and set up an MRM method, and we are now validating it under GMP. And it's the plan that from next year we should run release of new batches and that they have more than 200 batches per year. So slightly different application than just looking at host cell proteins, but it's still very critical to monitor, and it's also in very complex samples that we do this analysis. So I think that's the take-home message here is that with the MRM method, we can pretty much monitor any proteins that you might need to monitor by mass spectrometry. So to sum up, I think it's very nice that we have the new USP chapter coming out because it is setting the direction for HCP analysis towards using mass spectrometry. I think it will more and more will see the benefit of using mass spectrometry, but also the regulatory authorities will point to this chapter and say, yeah, please see how you can do it here. So if you're not using mass spectrometry today for monitoring your host cell proteins or residual proteins, I think you should get started soon. We have now this LCMS capability within the lab where we can do it under non-GMP or GMP validated assay. And as I mentioned, we can of course look at host cell proteins, but you can also, if you have other proteins, residual proteins or viral proteins, yeah, we can establish the method for those proteins as well. And I think LCMS offers a particular advantage for complex products, especially if you have short timelines. And I also think a lot of you with complex products will have issues with developing good ELISAs because typically you not only have one expression organisms, you might have 2 or 3 and use different types of growth media, and so it's going to get quite complicated to set up an ELISA or a number of ELISA assays to monitor those proteins. So that's the good thing about mass spectrometry. It doesn't care where the proteins are from, as long as you set up the right database and you select unique peptides, then you can monitor any given protein that you might have in your sample. And I think I also what I just mentioned about using it as a platform assay, I think if you have a platform for production of a vaccine or a cell or gene therapy or even monoclonal antibodies, we could actually validate the method for one project or for a few projects, and then you would show that this was an assay that you can use for your platform and for future projects, so that you would speed up the process even more and make it possible to use the assay for all new projects. With that, I hope you have enjoyed the presentation and got an insight into how you can use mass spectrometry from looking at complex products, and how the possibilities with the GMP accreditation now makes it possible for you to use it all the way through both early stage phases, but also phase 3 and for market. And then I hope you have some questions that we will try to answer. Thank you.
Thank you so much, Thomas, for a really nice presentation. Hope everyone enjoyed it. And I see we have a bunch of questions, so I'm thinking we'll just get right into it. But if you have any questions, those of you that haven't put it in the chat, be welcome to do that. We'll catch up on as many as we can squeeze in. So the first one I see here, do you generate the database for DIA analysis from a theoretical spectral library, or are DDA runs used first to generate the library?
Yeah, we always generate the spectral library on your samples or the samples for this project. So we run samples both doing DDA and DIA to set up the assay for those specific samples. And sometimes we just have one sample and then we set up the assay based on that, but we always run the DDA in triplicates to make sure that we find all the peptides and we add those to the iron library. So it's not based on previous data from other projects related to the same organism.
Okay, great. How many HCPs are you using for quantification, and do you use peptides or synthesized HCPs for quantification?
Yeah, we always use intact standard proteins, because we think that by using peptides, you don't take into account the sample preparation, so you don't take into account the efficiency of the digest and the matrix effect on that. So by using intact protein standards, we can control this, as I showed, this variability that is part of the sample preparation. So always intact standard proteins. And there was a second part of that question.
Yes. I think I lost the question now. Yeah. No worries about that. But I think especially for MIM, it depends. If you have the HCP or the residual protein recombinantly, I guess you would use that as a standard to do.
Yeah, that's used then for spike in experiments and showing linearity and lower limit of quantification.
So I think the first question was, how many HCPs are we using for quantification? I guess that depends on the product.
Yeah.
How many peptides do you consider for quantification? Also for MIM, how many transitions do you use for each protein?
Yeah, we normally use between 1 and 3 peptides for quantification, and we have 3 transitions. Then we have some peptides that are just qualifying the instrument. They are not used for quantification, but just to qualify the assay. But it depends a bit on the proteins, how many good peptides we can find. And also the assay, if we have the recombinant protein or in-house and can show that there's good linearity with those peptides we select, then we can go with just 1 or 2 peptides.
Yeah, I think this question is related, so it's sort of in the same direction. How is quantification being done? Do you have standards for each protein peptide of interest? Because the internal standard protein would not fly the same as all of the other proteins. So I think it's a mass spec expert here.
Yes, I think I think the assay, you're right about that different peptides fly differently, but I also want to say that by using the intact standard proteins for normalization, we still have a very good precision, even though the accuracy might be off by double or half what we typically see. Some proteins are even worse than that, but it's still very precise. And I think that's the main purpose with this assay, is to be able to compare from batch to batch and show the level of HCPs in the batches you are producing, and show that it's the same or similar to a reference sample. So I totally understand, if you want to know exactly the amount or the accuracy of the assay, then you need the recombinant protein for that specific protein, and you need to then do this dilution linearity studies or dilution studies, where you show that how the signal intensity correlates with the amount you have added.
Yeah. Thanks. When HCPs to be quantified are more than 50, what would be the approach do you suggest for quantification?
Yeah, that's of course the MRM method is not. It's suitable for, I would say, up to 50 proteins in one assay. Of course, you can then run the same sample with different methods and then add up the HCPs. But typically, we would then either take the top HCPs that are covering 70-80% of the HCPs. So if we analyze a number of batches and we find the top 10-20 HCPs, we can argue that they are covering 70 or the majority of the samples or the HCPs found in the samples, and then we would look for those. I would still recommend to run the same samples with the characterization assay. So as you saw, the sample preparation is the same, so we can easily run them both on the SWOT instrument and the MRM instrument, and then you can actually keep an eye out if the level of HCPs are suddenly increasing from some HCPs that are not in your MRM assay.
Okay. And then maybe you would even choose to do a non-GMP assay validation of that, like for case one.
Yes, and we are also working towards getting that assay up fully validated. But we just have a new instrument and we have a new software, and hopefully we will get there within the next year and a half.
That's going to be interesting to see. If switching from ELISA to MS, any problems with bridging? Also, typically WHO expect a limit of around 100 ppm. Does that type of limit make sense when using LC-MS?
Yeah, I think it's important to say that, and the regulatory authorities, they know that this is two different methods of monitoring host cell protein, and even ELISA it's not quantitative either. It's so with mass spectrometry you might get a lot of different results, but it might also be the same as your ELISA. So I don't think you need to do bridging. You should just show that the HCPs that you detect are correct, and that you have that your HCP assay is qualified and validated.
Yeah. Okay. What are the main differences between HCP analysis of a mAb product versus a complex product, such as a vaccine or gene therapy? I think it relates to the topic today.
Yeah, I mean, of course, as you know, most mAb products are quite clean, and you already have, for most of the mAb, they are typically expressed in CHO, so you have good ELISA assays. And we actually seen a number of our clients now with the ELISA MS coverage analysis that they can actually use an ELISA, commercially ELISA assay all the way through, even for market. So I think for those products, you should use the mass spec analysis to look at these lipases that might cause you issues even at very low levels. So it's more looking at critical or HCPs, but I will still stick with the ELISA assays. But for the more complex products, you have an issue that you can't use ELISA assays, and I think that's where and that's why we are giving this talk today. Is it's so relevant for people that have issues with an ELISA, and they still need to monitor their host cell proteins, and they still need to show that the batch-to-batch comparability. And with that, I think mass spectrometry is the only way to go. That's the MS only strategy, I would say.
Yes. Yeah, we still have a few questions on the quantification part. What are the quantification differences between SWOT and MRM? I think you mentioned it over the presentation, but maybe just sum it up briefly.
Yeah, I mean the SWOT and we are using an instrument from the same vendor, and actually this is really good because the data we get from the SWOT analysis are very similar to what we get when we look at the MRM data. So the best peptides we see in the SWOT analysis is still the best one when we are doing the MRM assay. But of course, the principle of the method is that with the SWOT, we have divided the whole mass range into smaller windows, and we do MS sequencing on all of these windows. And with the MRM assay, we look specifically for certain peptides, and then once we see them, we fragment those, and then we monitor the fragments with the MRM method.
Yeah, so it's almost like untargeted versus targeted approach.
Yeah, it is. It is. Yeah, and the beauty about the SWOT method is that you can always go back and look for new things you might not have seen first, or if you suddenly think that there might be a contaminating protein that is not in the database, you can always go back and research the data to make sure that it's there or it's not. So that's of course not possible with the MRM method. There you only look for what you target at.
Yeah. So we're almost running out of time, so I think we'll just take one or two questions more. Sorry, I may have missed, for MRM quantification approach, are you quantifying all the HCPs against internal standard proteins similarly to SWOT?
Yes, so we still, if we don't have recombinant HCPs, we will still use the standard proteins to normalize against, to take into account all this variability. And then accuracy of that assay might be not, I mean, as I again mentioned, it might be within this double or half amount, but the precision is still very good. So we still have very good linearity between when you look at a specific HCP and the signal intensity from that specific protein. So I still think it's a very good assay when you want to compare batch to batch, although the accuracy might be not as exactly as it is when we have recombinant proteins where we can do spike-in experiments.
Yes. So I think that's almost it. Have you compared internal protein standard approach versus relative abundance approach? If so, how comparable are they?
Yeah, we haven't done that yet, and I think one of the ideas about building up this database with thousands of proteins from hundreds of projects, I think it makes it possible for us to look at a lot of these questions, and I hope we will have time to include those questions when we start mining the data and seeing how the proteins behave when we use different ways of quantifying proteins. So I hope in the near future that we can present more data on when we mine this database.
Great. So I think we almost got through the questions, but if you have any questions out there that we didn't answer, please feel free to contact Thomas or me or just AlphaLISE in general. So we just got a new website, and so please go there, alphalise.com, and request or put in your questions into a request via the contact form there. You're always welcome to do that. But I think we'll just end on a last thing that I was thinking that, Thomas, if you had any last advice for those companies developing complex products such as vaccines, cell and gene therapy, spectrophage products, what would be your advice regarding whole cell protein analysis or residual protein analysis?
In general, I think you should start early on. That's also very nice with mass spectrometry, is that we can start anytime. We are not you don't waste time looking at samples, even though your process is not settled or anything. It's still the same assay can be used all the way through, even if you make major changes. As long as we know what proteins might be in there, then you can use mass spectrometry. And I think it's important to start early on so that you get a feeling for the robustness of your procedure, and you understand your product and also the impurities you have in there. I think the FDA said that you should do something, monitor something, just anything you can, and mass spectrometry is the perfect tool for for this. So I would just get going, and also with the new chapter in the USP, I think you will get if you don't do any mass spec, I think most people will get the feedback from FDA and EMA that they should do some type of mass spec analysis on their samples.
Yes, great. Thank you, Thomas. I think we'll end here. We're a bit over time, so let's call it here. But thank you all so much for joining this webinar, for your really nice questions. I hope you enjoyed the presentation. And if you have any questions, please reach out, and we'll be happy to get in contact to you and discuss any in this topic or anything related to protein analysis. So thank you so much for joining today. Have a nice day.
Thank you. Bye-bye.